of investigators and neurologists working on MS across the province. And our main goals are to provide world-class training to the next generation of researchers, and also to foster collaboration and networking on MS across the province. Just to acknowledge our steering committee, that we have Penny Smythe, Chris Power, Brad Kerr at the University of Alberta, Shalina Usman, Ketayun Alikani, and Scott Jarvis at the University of Calgary, and Dr. Galinda Metz from the University of Lethbridge. Normally once a year, at the end of the year, we would meet in Banff for an overnight retreat to network as well as to rejuvenate one another. Last year we did not do that because of the triannual NMS conference being held in Calgary this December, where we were scheduled to host one. I don't think we are going to be able to do so. So hopefully we'll meet next year sometime for the retreat. Okay then, on to the topic at hand, Bruton's tyrosine kinase inhibitors in MS. Just going to put on the laser pointer and move on to this next slide. To indicate that when one looks into clinicaltrials.gov, which is this database of clinical studies occurring around the world, and look into trials of Bruton's tyrosine kinase inhibitors in MS, one sees currently 5 clinical trials that are listed, all in phase 3, which is the definitive stage. We have 2 trials enrolled for relapsing remitting MS for evobrutinib, which is a drug by Merck EMD Serono. The trials are called Evolution 1 and 2. All of these trials have very fancy names attached to them. And also from Sanofi Genzyme, this molecule SAR442168 that does not yet have a chemical name. And it lists 2 phase 3 trials that are already recruiting. These trials are Gemini 1 and 2 for relapsing remitting MS. And there's 1 phase 3 as well for non-relapsing secondary progressive MS called Hercules. And I know that there are others to be registered as well. So with these number of phase 3 trials coming along, clearly targeting BTK is an emerging strategy, an interesting strategy for therapeutics in MS. But to this point, if you check PubMed, there are not many papers on BTK in the context of MS. So I thought that it is timely for us to come together to talk about what is Bruton's tyrosine kinase, abbreviated BTK. And so the flow today then is to begin with a brief reminder of the key disease states of all phases of MS, and then introduce you to BTK, and then tell you about the relevant published report on BTK inhibitors for MS, and then just address some of the gaps of knowledge for BTK inhibitors in MS. Just one, to declare my disclosures that as the director of the network, I reach out and obtain unrestricted educational grants to support the network from Biogen, EMD Serono, Novartis, Roche, Sanofi Genzyme, that I consult with these companies here, and that the current seminar is sponsored by Sanofi Genzyme. So beginning with the first portion then, a brief reminder of the key disease states of all phases of MS, which is relevant to thinking about BTK. So to begin then, this is a very simplified overview of MS pathogenesis, very simplified because I know we have undergrads in our midst and some of them are quite new to MS. So that in the healthy situation, we have immune cells that very few of them, but some do, make their way across the blood-brain barrier into the CNS for immune surveillance. That in people with MS, that there is overactivation of some pro-inflammatory immune cell subsets. They gain the capacity to cross the blood-brain barrier, and they then destroy elements of the central nervous system. Moving on to this more finesse description of MS immunology, this is a traditional view of MS pathogenesis. For many years, the condition is T-cell centric, that T cells activate macrophages, T cells activate B cells, that B cells are relatively passive players, that B cells mature into plasma cells that produce antibodies. So that for many years, traditionally, the pendulum then has been in favor of T cells, and that there is an imbalance where the T helper 17 pro-inflammatory subset outweighs the regulatory anti-inflammatory subset, and the situation is one of pro-inflammation. So that's the traditional view. And more recently, the view has changed to the collaboration of all of these cell subsets. The T cells activate B cells. B cells can themselves be antigen-presenting cells that activate T cells, that they're not mere producers of antibodies, but that they also release cytokines and other molecules. That B cells also activate macrophages, as do T cells, and that there is this tri-directional interaction. And so now we tend to think of pro-inflammation, but also a variety of immune cell subsets, not just T cells, but B cells and macrophages as well. And just to make a point about the importance of B cells, some of our more potent MS therapeutics are directed at B cells. These are the anti-CD20 monoclonal antibodies. And I highlight here this review, which is penned by a senior author, Amit Baral. He is at the University of Pennsylvania, very well known to many of us working in MS. And 30 years ago, he was actually a summer student in my lab. So this just tells you how old I am, but it's also to inspire the summer students and the graduate students amongst you that someday you will also be leaders in the field of MS. So this slide is to emphasize that lymphocytes continue to be important throughout all phases of MS. T and B cells are important very early on at the earliest manifestation, but they continue to be very important in the progressive stages of MS. An example is from this study by a UK group showing that in brains of patients with progressive MS, in the meninges as well as in the parenchyma, they can find aggregates of B cells labeled with anti-CD20, T cells labeled for CD3. So you can appreciate that there are many of this. And in fact, in this study, they subcategorize patients according to the density of these cells. Some have few of this, some have more, some have abundant. And with those progressive MS patients with abundant amount of T and B lymphocytes, these patients also tend to have a shorter lifespan as well. So I want to emphasize this because it is often thought that in progressive MS, inflammation has largely subsided, but indeed that's not the case. lymphocytes, as well as myeloid cells that we'll see in the next slide are very prominent, and they continue to be very prominent in progressive MS. So here's the involvement of microglia macrophages in progressive MS. Again, they can be detected from the earliest stages, but this shows us their substantial activation in progressive MS, that in relatively normal areas in primary progressive MS, these clusters referred to as microglia nodules are common and very common in progressive MS autopsy studies. Is this so-called expanding lesion. We're looking at a myelin state. Here is an area of demyelination. Here is still relatively normal, so the lesion is being pushed out this way. And in this border, one sees microglia and macrophages. They're called smoldering lesions because often one can follow these on MRI in living individuals, and one can see that these lesions are slowly expanding over time and correlating at histopathology, at autopsy, is this density of microglia macrophages at this edge, as if they were providing the toxic factors that help to drive and expand this lesion. And I want to emphasize that current disease-modifying therapies are not directed towards the CNS intrinsic microglia or the monocyte-derived macrophages that have entered into the central nervous system. And I've lumped them together as microglia macrophages because often at histopathology it's difficult to tell them apart. From this study from the Hans Lassmann group in Vienna is the emphasis that in progressive MS cases, not only is there a lot of microglia macrophage accumulation, but that these myeloid cells, microglia macrophages, bring with them producers of oxidative stress. So many of these microglia macrophages express NADPH oxidase, which is substantially upregulated compared to the normal situation. And this, of course, is an enzyme that produces superoxide anion, which is a free radical. Free radicals are very reactive and can destroy things in their vicinity. We also have iron deposition, another source of free radical. For many years, oxidized phospholipids are taken as an indication that oxidative stress has occurred because free radicals would oxidize lipids, so that the presence of these oxidized phospholipids are indications that oxidative stress has occurred. But Jeff Dong from my lab, a postdoctoral fellow, has a paper in Revision at Nature Neuroscience to show that these are actually very neurotoxic and continue to expand the injury. And in the midst of all of this are signs of axonal injury. In this review that Jason is a co-author on, we reviewed progressive MS and we emphasize that in terms of treatment approaches, we need to target B and T cells because they continue to be quite dominant. Even if they are not aggregated in meninges or perivascular space, one can see them distributed diffusely across the parenchyma. And of course, we still need to target microglia macrophages. These are within the central nervous system, so we need drugs to enter into the CNS. And other desired actions would be neuroprotection and also remyelination to reduce the axonal loss that contributes to progression of disability. So with that disease state overview, we now go into the next segment of Bruton's tyrosine kinase. So this has its origin from this description by a pediatrician, Colonel Bruton, working out of Walter Reed Hospital in Washington, in which he described a young boy who was highly susceptible to infection. And this boy had the complete absence of gamma globulin in human serum, which we now know to be antibodies. So this boy could not make antibodies. And Colonel Bruton charted the infections that this boy encountered. So over just a few years, 1946 to 1951, look at the number of sepsis episodes that this boy had to endure and other infections, other diseases. Here's the treatment that he administered. And eventually, when a gamma globulin fraction, meaning antibodies, were infused into that patient, that boy recovered. So this was the first description of individuals without any antibodies, A-gamma globulinemia. And the story then unfolded some 40 years later. Two groups, one in Science, one in Nature, described the gene mutation for that condition, and they named the gene BTK, or Bruton's tyrosine kinase, in honor of Colonel Bruton. They renamed the condition X-linked A-gamma globulinemia, or XLA, because this gene is found on the X chromosome. So patients with this gene mutation lack antibodies, but they do relatively well when IVIG, intravenous immunoglobulins, is infused, so providing these patients with antibodies. From a number of studies now, we now know that BTK is expressed in all hemopoietic cells except T cells, interestingly enough, and that it is downregulated in plasma cells. It's important because when one uses BTK inhibitors, the expectation is that one would be sparing plasma cells that can still produce antibodies. So this slide shows the development of B cells that occur from a hemopoietic stem cell in the bone marrow. Within the bone marrow, there's subsequent successive maturation to pre-B cell. It then emerges from the bone marrow, goes into secondary lymphoid organs for further maturation. And one of the end stage cell is the plasma cell, the mature B cell that secretes antibodies. With regards to this mutation results in this termination here, where patients cannot go beyond this stage, cannot make pre-B cells, and so subsequently you have deficiency and therefore the inability to make antibodies. With the use of BTK inhibitors, which do not deplete BTK but inhibit the BTK activity, that one would expect that there might be some reduction, although that's still unclear, but certainly as we'll see later, the expectation is that these cells are inhibited, at least in maturation, development, and also in function, without necessarily depleting the number of B cells. So this is where the BTK biology was first systematically studied in B cells, and in response to cross-linking of the B cell receptor on the surface of B cells that lead to the activation of B cells. So here we have an antigen cross-linking the B cell receptor, initiating the activation of a number of tyrosine kinases that then go on to phosphorylate and activate BTK downstream. So phosphorylated form of BTK is taken as an indication that BTK has been activated. Following activation, because it is a kinase, BTK goes on to phosphorylate a number of downstream targets, including PLC gamma 2. This is important because it generates two important secondary messengers within cells, IP3 that mobilizes calcium from internal stores, diacylglycerol that goes on to activate protein kinase C and other events, and this lead to the activation of transcription factors such as NF-kappa B, leading then to the production of pro-inflammatory molecules. So BTK then mediates signaling within B cells, and there's some thinking that it could be the rate limiting step. It is a funnel point, it is a control point, and if you inhibit this BTK activity, you are going to inhibit downstream signaling, even though the upstream receptor has been engaged. So it's important for B cell development, function, and survival. At least from the rheumatoid arthritis and lupus literature, where autoreactive B cells are found, and where these have upregulated BTK activity, these autoreactive B cells seem to depend upon BTK for survival to a greater degree than normal cells. So in those autoimmune conditions, there is anticipation that BTK inhibitors would be more effective in reducing autoreactive B cells, while perhaps sparing some normal unactivated B cells. May promote autoimmunity. Levels of BTK is elevated in peripheral blood cells of patients with rheumatoid arthritis and lupus. And if BTK kinase activity is dysregulated, overactivated, this gives rise to B cell lymphomas. There are transgenic mice that overexpress BTK, and this is BTK overexpression in B cells. And now when these B cells are stimulated, one sees that there is a greater calcium flux, greater degree of activation compared to normal B cells from wild-type animals. There is a mutation of BTK in mice. This is the XID mouse. And in this mice, the development of EAE, which is experimental autoimmune encephalomyelitis, an inflammatory model of MS, is attenuated. So while EAE occurs in wild-type animals, in mice that have BTK mutated, it's harder to produce EAE disease. This authors also went on to study XID BTK deficient macrophages in culture, and these produce less reactive oxygen species, leading to another connection in MS, if you like, that if BTK is inhibited, there would be less reactive oxygen species, less free radicals. So BTK, while first described and focused upon in the context of B cells, has really taken another gear in the context of MS with the discovery that BTK is also very important for macrophages microglia. So BTK signals downstream of many receptors on myeloid cells, such as this, including toll-like receptors, that BTK promotes NF-kappa B pro-inflammatory signaling, formation of free radicals in these myeloid cells, that BTK leads to inflammasome activation that generates pro-IL-1 beta. And I have a schematic here to show you macrophage with various receptors on its surface, FC receptor, etc. BTK involved in the signaling cascade through many of these different receptors, that the activation of this inflammasome is also modulated through BTK. And so therefore, if this is inhibited, the expectation would be less pro-inflammatory activation, including the reduced production of a pro-inflammatory cytokine, IL-1 beta or IL-6. Okay, I meant to show you this other point, but it has skipped this a couple of slides. Let me see if I can regain those. Oh, actually, it skipped. Okay, here it is. Another point about BTK within macrophages. There is a published report that in macrophages from lymphoma patients treated with a BTK inhibitor, ibrutinib, that these macrophages increase their M2-like expression. And I just want to tell you about M2. So here is polarization of macrophages or microglia. This shows you that these myeloid cells can appear in a spectrum, that there are many different forms or phenotypes, if you like. And here we are showing two of this, the so-called M1 pro-inflammatory, which is generated when one uses activators such as LPS and TNF, that these pro-inflammatory macrophages then produce IL-12 to activate pro-inflammatory Th1-17. So this M1-like is very pro-inflammatory. But you also have another type or another phenotype that is M2-like, that this is generated by these activators here, including IL-10, that the M2-like cell goes on to produce cytokines such as TGF-beta, IL-10, that then generates Th2 that tends to be more anti-inflammatory. So the M2-like cell is referred to as homeostatic or regulatory macrophage microglia. In MS, there tends to be more of M1-like, less of M2-like. And we have in our lab been thinking about how to generate M2-like cells for a very long time. But it appears that the BTK inhibition may actually fulfill that role, that may promote a homeostatic M2-like phenotype. So besides being present in myeloid cells where BTK may mediate the activation of a pro-inflammatory phenotype, the inhibition of BTK may also generate a regulatory, potentially reparative macrophage microglia population. So BTK inhibitors then would hopefully normalize adaptive, where it was first described on B cells, but also innate microglia macrophage immune responses in MS. So I want to go back to this slide that seems to have skipped. And this is a slide that was recently presented at this canceled meeting, the American Academy of Neurology. Nonetheless, the presentation was later made available. And here they were sampling MS tissue. Here's a demyelinated area where in this area of demyelination at the edge are cells that upregulate BTK expression, and where they were identifying the type of cells that were expressing BTK in that edge of that lesion. They found that most of these were IBA1 positive, meaning that they were microglia macrophages. So in MS lesions, in an active area, there's BTK upregulation in microglia macrophages. They went on to document the expression at the level of protein and mRNA versus healthy controls, documenting that at least at the level of protein by Western blot, they can see more upregulation in primary progressive, although this seems elevated in secondary progressive as well. Their RRMS, relapsing remitting, did not show that elevation, but this seems to be the case at the level of transcripts and across the different forms. And this is just to emphasize that BTK, therefore, is upregulated within the brain of patients with MS. Okay, so this then brings me to the next segment, which is with the upregulation of BTK, at least in lesions in the brain, and with the biology that BTK is important in B cells as well as in myeloid cells, both of which are important for the pro-inflammatory characteristics that we see in MS for immune cells, is there a role for BTK inhibitors? But first, an introduction to the BTK inhibitors. So this is what they look like. Here I'm presenting to you three BTK inhibitors. This is fenebrutinib, which is a BTK inhibitor that Roche has used in rheumatoid arthritis. This is the Sanofi Genzyme compound. Here is evobrutinib, EMD Serono's compound. These two are in clinicaltrials.gov at this point for trials, phase 3 trials in MS. Here are a few other BTK inhibitors, and we can see that they are polyphenolic. They all show some similarity to one another. Ibrutinib is one of the leaders in this area, and it was, in general, BTK inhibitors. Ibrutinib being the first, is approved by the FDA for use in lymphoma since 2013. When one looks into clinicaltrials.gov, this is actually a fairly active area. A week ago, I had 167 entries. This morning when I looked, there are 187 entries in clinicaltrials.gov, this database of clinical studies around the world. So 187 entries. Most of this, though, is in the context of various B-cell malignancies to inactivate B cells. But some of these are also in rheumatoid arthritis, in Sjogren's, in lupus, graft versus host, and as of today, 5 phase 3 trials in MS, 2 with evobrutinib and 3 with this Sanofi Genzyme compound. With regards to biology, here is an experiment in tissue culture where B cells were activated by cross-linking their B cell receptor and then looking at an activation marker. So here's the resting B cell in culture, now activated with activation marker going up. And in the presence of this BTK inhibitor, the activation was subdued. These tend to be irreversible covalent inhibitors, so that when the inhibitors, in this case, the inhibitor was washed out, that inhibition was retained. This is just to demonstrate that T cells are relatively resistant to BTK inhibition, where when T cells were activated, and again, activation marker displayed here with increasing concentration of BTK inhibitor, they continued to be activated.
I didn't get that. Could you try again?
Sorry about that. Here's looking
No need to apologize.
Here's looking at a microglia line. I was trying to look at my time to see how much time I have. So here's BTK in a microglia cell line, the BV2 microglia cell line, activated now with LPS, upregulating a number of pro-inflammatory cytokines such as TNF, IL-6, IL-1, upregulating INOS that produces nitric oxide as a free radical. And in the presence of ibrutinib BTK inhibitor, one abolishes that. And here's the quantitation shown below. Here's another experiment, and in this case with an MS-relevant BTK inhibitor. Here's using mouse microglia, which is now activated. And as I've indicated earlier on, the phosphorylation of BTK is an indication of its activation, so that with the inhibitor now being present, that inhibition is of activity is manifested without affecting the protein level of BTK itself. In this study, the authors activated microglia, and they show that there is a lot of genes that change. This is an RNA sequencing experiment of mouse microglia. And after activation and in the presence of a BTK inhibitor, in this case, the Sanofi Genzyme compound, the RNA sequencing data seems to be normalized, or at least relatively close to that seen in controls. And it went on as well to identify a number of pathways that were activated by stimulation of microglia that is BTK inhibitor responsive. Here's the listing here, mostly in the form of pro-inflammation. But one of them that they highlighted is this gene RGS1, because this is the gene implicated from the GWAS study as a gene defect in microglia that increases the risk of developing MS. And they show that when microglia is activated, this goes up, and in the context of BTK inhibition, this is reduced. Other relevant thoughts about BTK inhibitors is the potential that by affecting microglia macrophages, that the consequence could be one of remyelination. Here's an experiment done with ibrutinib. This is the EMD Serono compound, and the authors used two models, transgenic tadpoles, where oligodendrocytes turn green when they are myelinating, or cerebellum slices that in culture, they can demyelinate by adding this toxin, lysolecithin. The slice subsequently remyelinates, and they are able to document remyelination by the use of markers, appropriate markers. And what they show is that BTK inhibition increased remyelination 1.7 times in the transgenic living tadpoles. And in these cerebellum slices from the mouse brain, the BTK inhibition following demyelination improved remyelination. So there's this anticipation that there might also be a remyelination outcome when BTK inhibitors are used. I should have mentioned that these BTK inhibitors are all CNS penetrant. And indeed, the Sanofi Genzyme study showed that in a phase 1 trial in which human volunteers were given their BTK inhibitor, that inhibitor can be found in the cerebrospinal fluid of human volunteers. So this brings me then to the available clinical results of BTK inhibitors in MS. And again, I refer you to this slide that I showed you earlier on of signaling through B-cell receptor engaging BTK, but a reminder that BTK is also important in myeloid microglia macrophages. The available data for MS at this point is this study by EMD Serono of ibrutinib. I'm going to show you a couple of data sets. This has phase 2 data. The Sanofi Genzyme compound also has phase 2 data, although that's not in the public arena at this point, but I do have a press release to show you. So this is the EMD Serono ibrutinib compound, the phase 2 study published last year in the New England Journal of Medicine. So basically then, the primary outcome is the number of gadolinium enhancing lesion over 16 to 24 weeks. I actually can't see the top of the slide, but anyway, they added the number of GET enhancing lesions over that period of time. This is a phase 2 was placebo controlled. So over that 24-week period, we have ibrutinib 25 milligram once a day or once a day at 75 milligram. And this produced a statistically significant reduction in a number of gadolinium enhancing MRI activity, an indication of inflammation in the brain. Their positive control was dimethyl fumarate. They had a problem in this study with dimethyl fumarate because the positive control did not work. They mentioned that one of the patient had a lot of gadolinium positive scan lesions that threw the data off. But this was promising, 75 milligram once a day. Twice a day also had a trend towards a reduction, although it was not statistically significant for the adjusted p-value. They also look at annualized relapse rate at week 24 and 48. One usually needs a larger study as well as a longer period. And here they showed a trend towards a reduction in annualized relapse rate, although that did not arrive at statistical significance. In that study, though, they reported that the drug was well tolerated. So when they looked at the common adverse events, looking at the placebo as well as drug treatment arm, nearly all of these were quite equivalent to that of placebo. I should say all of these were equivalent. And as we go through this list, they concluded that this was a drug that was well tolerated. Here's the Sanofi Genzyme press release in which they did a placebo-controlled phase 2B study of 12-week treatment with this compound, where the primary outcome was, again, the number of gadolinium-enhancing lesions, where they showed that the 60 milligram dose reduced the number of GET-enhancing lesion by over 85%. And in terms of the secondary outcome of new or enlarging T2 hyperintense lesions, there was also a prominent reduction. They described that the treatment was well tolerated, that the most frequent adverse events were headache, upper respiratory tract infections, but all of these were quite close to what the placebo arm was showing. And in that press release, it was noted that Sanofi is moving rapidly to initiate 4 phase 3 clinical trials. As of today, 3 of these are registered in clinicaltrials.gov, all phase 3, 2 of them in relapsing remitting MS, and 1 of them in non-relapsing secondary progressive MS. So there are still many gaps of knowledge, though, with regards to BTK and BTK inhibition. And I'm going to list a number of this here. Efficacy in MS animal models. There's not a lot of published data at this point, although I think that this data does exist in the EAE model, but not yet in the published arena. Long-term efficacy in MS, that remains to be seen from the phase 3 studies that are going to be conducted or have been initiated. Long-term safety, a question mark. What about B cell and plasma cell counts? The expectation is that the counts would be relatively normal, that these cells are just inhibited rather than depleted, that the anti-CD20 monoclonals do with regards to depletion. Here, the BTK inhibitors are thought to produce inhibition rather than depletion of cell numbers. But is that going to be the case with long-term usage? and with long-term usage leads eventually to reduction of immunoglobulin levels, even though BTK is not thought to affect plasma cells. What about oligoclonal bands in the cerebrospinal fluid that are reflection of immunoglobulins of various specificities? Would this be reduced? We don't know yet at this point. What about effects on other leukocyte populations like T cell counts, neutrophils, etc.? We don't have that data yet, or at least not in the published arena. Is BTK or the active form elevated in MS? Well, some evidence of this being elevated in MS lesions, in microglia. The whole story is yet to unfold, I think. And what about cells in the periphery? Do B cells from MS individuals have elevated BTK activity during a relapse? We don't know that at this point. Would these inhibitors lead to neuroprotection by virtue of their capacity to reduce free radical generation, at least as demonstrated in tissue culture? Would they actually inhibit or induce microglia to become M2-like homeostatic, reparative, anti-inflammatory state? Don't really have that information at this point. While there are indications that there may be remyelination in the transgenic tadpole and tissue slices models in culture, will there actually be remyelination in animal models, let alone patients with MS? So these are all questions to be answered. So a summary then of the emerging therapeutics around BTK inhibition, that these are of interest and some excitement because they seem to produce B cell paralysis rather than depletion. And they also affect macrophages, microglia, a two-arm approach, that they may inhibit the pro-inflammatory type, may generate the regulatory reparative type. They do enter the central nervous system, and whether neuroprotection and therefore reduction of disability is going to be an outcome remains to be seen. The potential of remyelination is certainly there, but we do need data in that regard. And here I'm going to show you this slide that my youngest daughter actually drew up. She's in third year, final year med school here at the University of Calgary. I'm very proud of her writing this review when her school was shut down due to COVID-19. I'm editing this at this point, but her emphasis was on what is required for a successful medication in progressive MS. So she put together this slide using BioRender software. A drug needs to enter the central nervous system because of all the inflammation, microglia activation, B cell, lymphocyte, T cell accumulation within the central nervous system. So the drug needs to get in, needs to neutralize this compartmentalized lymphocytes, need to antagonize or inhibit the pro-inflammatory activity of microglia macrophages, need to be able to detoxify free radicals, promote remyelination, hopefully exert some direct neuroprotective effects. And at this point, from the limited literature and certainly the hype that is available, I would say that the BTK inhibitors have a promise actually to be able to meet all of these marks. So I'm excited about where the BTK inhibition field is going to go. So with that, thank you very much for listening. And thanks also to Valerie and Sanofi Genzyme for sponsoring this talk. And I just want to leave you with this last slide to thank the people in the lab who are always the ones doing the work. that expand our knowledge. So thank you very much. And with that, I'm going to try to see if I can now get to the comment box. Okay, so let's see.
We don't have any questions,
No questions? I am ready to address questions.
Hi, this is Katya Yong. Thank you for the great talk. I can ask a question. So the BTK inhibitor that is approved for lymphoma, ibrutinib, it is associated with some cardiac toxicity, including atrial fibrillation clinically. So I was just wondering if this is, what the I haven't really looked deeply into this, but what could be the mechanism of this? And was there any concern in the clinical trials with cardiac toxicity in MS patients?
Not to my knowledge. I have to say that I did not look at the literature around ibrutinib. And that's because it is not one that is being pursued in MS. Somewhat of a first generation BTK inhibitor. So I did not look into its side effect or the potential of the newer generation BTK inhibitors producing cardiac issues. The literature that I've looked at with ibrutinib, with the Sanofi compound, and also with fenebrutinib, which is a Roche compound that has a phase 2 study in rheumatoid arthritis published. I did not come across cardiotoxicity. That's not to say that I just have not seen that, Katya.
Thank you. It was a great talk. Thank you.
Hi, this is Fabrizio speaking. Can you hear me?
Fabrizio, yes.
Hi, So thanks again. It was an excellent talk, a very good overview of these new compounds that, as you mentioned, I mean, they can be quite exciting because of these almost double action, one on the immune system and one also within the CNS. So this could be kind of very interesting. One of the questions I have is about the effect they have on the B cells. So you were talking about this B cell paralysis, almost like, the result of these treatments. And do we know if there is a differentiation on the effect that they have between the antigen presenting abilities of the B cells versus instead the other function that is, generating plasma cells and immunoglobulins? The reason why I'm asking this question is because obviously in the field now of the B cell depleters, there is more and growing concerns about, the immunoglobulins levels as they seem to decrease over time. And obviously that can have two effects. One is on the susceptibility to infections, and the other one is obviously response to vaccinations. So can you comment on that? Thank you.
Yeah, Fabrizio, those are all great questions. I've been trying to get the data. I'm trying to find the data, and I've not been able to find the data at this point on, for instance, from the ibrutinib trial, they must have collected blood and leukocyte populations to try to get at some of these questions, but I am very keen to see the data. Is there a reduction in B cell counts? Is there preferential action on cytokine production versus antigen presentation? Immunoglobulin levels, are plasma cells spared? At this point, there's a literature around the downregulation of BTK levels in plasma cells. So potentially they are spared, but as you know, plasma cells are generated from B cells that have been activated and that mature into antibody secreting plasma cells. So will that have an impact long term? Don't know. One exciting feature for me at this point is the potential that we are just inhibiting, at least for some time, rather than profoundly depleting, B cells. Might that be the differential between anti-CD20 depleting monoclonals and these BTK inhibitors? So I think there are still a lot of questions that are unanswered at this point. I looked into the lymphoma literature, but the lymphoma literature and MS literature could have different outcomes. Because those are conditions in which there is a defect in central memory in B cells in the central compartment, whereas MS is thought to be more, perhaps peripheral tolerance abnormality out in secondary lymphoid tissues. So I don't have the answers for you, and I think that there could be some data sets that I'm not aware of that are not published. But those are all questions that I have as well.
Thank you.
Is there another question?
We have more questions in the chat box.
Oh, sorry. Oh,
Yeah, go ahead.
this is Hedwig. Thanks so much for that talk. It was very informative, very good overview. I think you can guess where this is going. Is there anything known about expression of BTK in astrocytes? And also I was wondering, is there a difference between white matter and gray matter expression of BTKs?
Wow, great question. So it's not in the published literature. At the American Academy of Neurology, I think the meeting was in April, but the meeting was canceled. But some slide decks were made available. I saw a presentation where actually Bruce Trapp and Sanofi Genzyme had collaborated on, in which the BTK expressing cells in the central nervous system were microglia macrophage. But I saw another presentation in which they estimated that about 80% of the BTK expressing cells in the CNS were microglia macrophages, about 20% were reactive astrocytes. The oligodelineage cells did not express BTK. And that's the available data that's the data that I'm aware of at this point. I haven't seen anything about white matter versus gray matter expression.
Great, thanks.
Yeah, so I see it's 1:02. I will bring this to a close then. I want to thank all of you for your attendance and for your support of the Alberta MS Network. So keep it up. Thank you all.
Thanks, Wee.